Welcome to the final day of the conference. Thank you, Dr. Wolf, for chairing today's session. You can go ahead.
Good morning, everybody. Welcome to the last day morning session number 15, setting the standards and roadmap for organ-on-a-chip models. Before Sandra starts, I would like to explain the rules again. So we will have 45 minutes in total for the presentation. There will be 35 minutes presentation and 10 minutes for discussion. I will give you a reminder, 30 minutes, and you have then 5 minutes left to close your session in time. So I would like to thank Sandra for giving the presentation here about good life science model methods and biodata practices. Before I hand over, I will introduce Sandra. She's a team leader and senior scientist at the European Commission Joint Research Center in Italy, ISPRA. She received a PhD at the Free University of Brussels. She has more than 30 years of experience in vitro toxicology, including research and activity related to the development and validation of new in vitro cell and tissue-based methods. She received the Björn Ekwall Memorial Award in 2020 in recognition for her achievements in the validation of new methods in the in vitro toxicology area. Dr. Coecke is also highly recognized for her leading role in the development of the European Commission Joint Research Center's guidance document on good cell culture practice and the OECD guidelines document on good in vitro method practices. Her talk will concentrate on the good life science model and methods and biodata practices. Welcome, Sandra, and the stage is yours.
Thank you so much, Armin, for this introduction. I think the longer you work in the area, the longer the introduction. I'm extremely pleased and I'm very grateful for Surat and the whole team, the organizers, for inviting me. I'm also so grateful that I meet now, unfortunately, hybrid, some of the colleagues like Armin, David, Thomas, that you have been seeing, and others, which have, crossed, my professional path, and probably also you will see we all became friends to work in this field. I think it's a field, like I put on my first slide, where I hope for those that work maybe already a long time in this area, or those that are, let's say, the next generation, that I can inspire you with my talk, which gets a little bit a personal flavor, just to show you a little bit the struggles that we went through in all these years. But our strong belief that by embracing science, technologies, innovation, quality, but also speed and efficiency, we might really start embracing the next generation methods for better understanding the mechanism of systems biology, the mechanism of toxicology, and also of efficiency of therapeutic interventions. So to give you this flavor, I started with, showing a little bit how I ended up in this field. Basically, I started education as an engineer, biotechnology, but already soon I got to know in the Faculty of Medicine a lab that was working on tissue engineering. In that time, it was, let's say, alternatives for skin therapies. We worked at a university in Brussels with a hospital that dealt with people that suffered from severe burning wounds. And so our lab in Brussels was helping to isolate biopsies of the small parts of their let's say, skin layers that were still intact to help them survive these severe burn wounds. And that's how I ended up in the area of tissue engineering very early in my career. So already in 89, I started to work, let's say, on this type of new biotech methods. Then, thanks to my professor, who was Vera Rogiers, she got in contact with the pharmaceutical industry in Belgium, Janssen Pharmaceutica, and they had been working a lot, let's say, with animal models. They had big tox department, but they had a small group that started to work and embrace some of the in vitro cell and tissue culture methodologies and some alternatives for getting a better grip on mutagenicity. So they had several methods, in vitro methods, to look, for instance, at the formation of micronucleus, etc. And I decided, thanks to Vera, to join this company who were looking to set up a lab of in vitro toxicology. So on this black and white photo, you see my team there in Janssen Pharmaceutica. I was very young, and I'm very grateful to my head in that time, Philip Van Parijs, who gave me already a lot of freedom to start up setting that lab. That has been a fantastic experience because, as you know, in pharmaceutical industry, before you put a compound on the market, you need to work according to some high quality standards. These are called good laboratory practice standards, and FDA, EPA, but also OECD uses these standards because it facilitates general data sharing globally. So when you work according to GLP and you work with a method where there is also a test guideline at the OECD, these data that are generated can be reused and you don't have to duplicate efforts. So in a way, this is already a kind of reduction of animal use when you do animal studies on the GLP. And my task was to start to see if I could collaborate with OECD to see if we could issue a document on in vitro GLP guidelines. And my track continued, and I started to coordinate and work a lot at OECD level over my career to embrace, let's say, these good cell and tissue culture practices or good in vitro method practices, as Armin as I was introduced. So basically, my whole life, I think I've been working to try to use these next generation systems biology methods to better be prepared and to better respond to health crises, to new diseases, but also to better understand the toxicology. And I show on this slide that actually I'm a mom of 3 girls. And my family is coming from a place in Belgium between Antwerp and Brussels, Willebroek, a very small place, where we have, let's say, where we had always a paper factory. And by coincidence, you know, my personal story links a lot to the work I'm doing. And I will show this by getting you through to, in a way, 3 decades, almost 30 years, we're all trying to standardize and characterize these cell and tissue culture methods. Because what we have learned, new methods, people need to trust them. So you need to have high scientific quality methods to be able to introduce new methods for also regulatory decision making. Now, these cell and tissue culture methods, although they are beautiful technologies and you can get so many answers from these methods, they have to be quite well controlled because small changes can impact the data you get with these methods. So I've been working a lot to characterize the critical elements in these in vitro cell and tissue-based methods, especially when you use them for regulatory decision making, but also for any kind of scientific applications. And furthermore, as I said, working with OECD or with other standardization bodies, like European Pharmacopoeia, we have been working also with Thomas Harting on some pyrogen methods. It helps for a global harmonization of these standards. And in a way, as I said, ultimately, why we are doing that. We want to be ready, and now with this fantastic EMBO course, to also use the latest technologies, so microphysiological systems, organ on a chip, for better response and preparedness for health crisis. One health crisis, because these methods can be used for human health, but also for animal health. And many of these methods, the principles can be used also for environmental health. So I've been working, in a way, 30 years in life science. And as I just mentioned, One Health. So I'm working now at the European Commission Joint Research Center. This is the research angle of the European Commission. We were very close also with the funding, where the funding comes from, DG RTD, research and development in that time. But we also work very closely with Santé. And, all things are linked. When you show this slide, human health, safety, efficacy, animal health, safety, efficacy, environment, plants, everything is linked. Food is linked. You see this little boy on the first slide. So for the last year, I've been quite a lot working for last years with our European Food Safety Agency and with our colleagues at DG Sante, so food and health. And I've been working on a document also together with the scientific board of, scientific advisors of the European Commission to issue a scoping document on sustainable food systems. And all what we're talking about here with these new methods boils down that for all these areas, for One Health, we need good methods, innovative methods. So One Health, this is a picture actually from me because I'm born, I'm living in Italy now since 96, but as I told you, I'm born in Willem, an industrial zone between Antwerp and Brussels. And coincidence in life is always, I loved chickens when I was a small child. I loved animals. So probably why I ended in this area to use better methods and also, let's say, to reduce the suffering needless of animals, because often what we saw also is that the conventional toxicology methods not always lead to answers we need to predict, for instance, why I get sick from a compound and others not. And I will show you that also with your slide. Chickens are quite important because I don't know, we don't have really an interactive format. I would have loved to be there with you, but as I already shared, because I was in contact with several of you via LinkedIn and Twitter, I could unfortunately not come because my little girl, I have 3 girls, one of them is now in the hospital, and actually she has been a while in intensive care in the last weeks. So I feel even more stimulated, with all the health issues my family has been having, my girls, So I think science is really a strong motivator to help, let's say, the people that I care for, but also, let's say, all the people and all the animals that would need her science. And this lady is for me, has been always a very important lady. Actually, she has been studying also, chickens in a way. This is Rita Levi Montalcini. She is one of the women that got the Nobel Prize of Medicine in 1986. And she got that together with Stanley Cohen. She got the Nobel Prize for psychophysiology and medicine, because they brought two distinct words together, chickens and research labs. And they discovered the nerve growth factor. And also in our work, and I think also in our Arwin work or David work's nerve growth factor, neurotoxicity, neurobiology, three-dimensional models are very important. So developing novel strategies is quite important. So I've been working 30 years, with chemicals and pharmaceutical projects, especially for toxicological assessment. So mechanistic toxicology is important. Quite early in my career, I taught that I started in the Free University of Brussels. So, working in these labs, this is a photo of one of the first collaborative project, a European Commission funded project, where I went to Marseille because we were trying to cryopreserve hepatocytes from different species, because we wanted to have liver cells cryopreserved from humans. And we started to see how the cryobiology that we learned from other species could help us to get better cryopreserved liver cells. What I also learned quite early is that in this field, to have some progress, you have to work together. So we started with the first conferences, horses, the Invictus Congress. Now it's called STIF. And several of you, I know, might maybe going to the STIF conference, which will be in Barcelona at the end of this month. So this was, let's say, one of the first STIF conferences that we organized with my professor Vera Rogiers in that time. I had a fantastic job that I had to look for the venue where we had our conference dinner. So I loved that job. But what I learned was that we need to collaborate in this field. So my whole life, I've been working with people in this field and connecting people for, let's say, amplifying our research results to get quicker these new methods in, for instance, OECD test guidelines. By a coincidence, when I was at the Janssen Pharmaceutica, I got into contact with this European Center for the Validation of Alternative Methods, which was led in that time by Michael Bowles. And one of the first workshops they did to bring people together was on the practical applicability of hepatocyte cultures in routine testing. I think this week you have been learning a lot on how important the liver is also for, organ on a chip technology, because it's our metabolizing factory. And so in that time, we started to look how we can get these methods standardized. And I also, as I told you in my beginning, I worked also on skin models and in vitro skin and eye irritation methods, corrosivity methods. And you could say how topical toxicity links to systemic toxicity, so target organ and target system toxicity. Actually, today, still, we see that these different disciplines need each other. We need to get a deep knowledge of systems biology, both topical effects, topical models, dynamics, and kinetics, to really deal with the complex diseases. I mean, I put on this slide, for instance, I leave you also publication. For instance, primary biliary cholangitis, it's one of the most common immune disorders that cause liver disease and primarily affects, by the way, women over the age of 40. And basically, you see that there is a big connection to what we know, what is happening, bile mechanism related, because this disease is characterized with symptoms such as chronic fatigue, severe itching, skin itching, for instance, dry eyes, and a lot of pain. So by having all this toolbox of in vitro methods together, we really can start, and we have been trying to use a lot of these methods also in biomedical research, not only in toxicology, which is the area that I've been most. But lately in my career, I start to to work a lot on diseases. So also I've been working also on pathogen effects, not only chemicals and diseases like COVID-19. So on the right slide, on the right part of the slide, I say I want to show that it's so important when you work with these models that we really work according to high standards. This is also the session today. And GLP, GCCP, GMP, you know, if you work in a clinical setting, in a pharmaceutical setting, you know, this is the way to go. So this is also the area that I've been trying to do. So I started to work in Italy in 1996, and that helped me a lot to help to get more cell and tissue models into OECD test guidelines. These are two of my girls. I have a third one. It's the third one that is in the hospital. But for instance, there, the big one, at 2 years old, she had already a skin cancer, because I'm coming from a very severe polluted area, many of my family. So, you know, I've been always trying to understand what is happening, you know. So understanding biology and systems biology helps us also better to develop methods. And by coincidence, you see there really live Armin Wolf. We worked already very, very early on together with people. This is 21 years ago. I showed you the first workshop on ECVAM. This was already workshop 45. This is kind of what we try to do as a bit of background, what you're doing in the MBOK course. We started to look how you can better, longer term, cells and tissues into culture. So, you know, I could say this a little bit. It was at the forefront of let's say, microphysiological systems. So I'm very pleased that Armin is now sharing this session, and that also you could enjoy, let's say, the incredible talks that always Thomas delivers, and we still collaborate now after all these years. Interesting is that I saw also that you know, in India, you do a lot of work of also embracing, let's say, these high practices now for in vitro studies. So I've been working with OECD. In 2004, we were able to issue this application of quality standards to in vitro studies. So that's a way where standardization and harmonization helps to boost this field. So you can, I leave you the slides also. This is some of my work over the years in topical toxicity, systemic toxicity. You know, you can find my publications and those of many of my collaborators. But what I want to show is, and I showed it already with this first example, everything is connected. So what we did in topical toxicity serves to understand, for instance, a systemic toxicity. And from the beginning, we have been using automation, omics, and other technologies to really get these methods really standardized and get the best and the most information out of them. What I also learned is that I saw you have some speeches on, you know, we talk a lot about personalized medicine, but, you know, why I'm doing this work is because we're talking also about personalized toxicology. And for instance, I myself and my friend Sylvia, we have been taking drugs, good drugs probably, you know, when they came on the market. But luckily there is pharmacovigilance, because sometimes, even if you do cohort studies in pharmaceutical industry, you might have adverse effects when compounds are on the market. You know, these are two cases. One is the classical case of thalidomide, where we saw adverse effects, you know, reproductive effects by thalidomide. And the other one, I did the experiment by speaking on myself in the small slide. You see myself here in 2015. I think I weighed only 35 kilos. I was severely intoxicated from an anti-leukotriene, montelukast, for my asthma. I took it chronically, 10 milligrams. So that means not everybody dies, because in this paper, actually science saved my life. The person that took it also 6 years, he died from drug-induced liver injury. I was also totally yellow. I had many systemic effects, but I survived it because I stopped the drug. And this drug had an adverse effect on me. So the difficulty of the science we're doing in toxicology is that not everybody, and also in pharmaceutical industry, but also in the chemical world, is that we have also individual effects. And thanks to all these new approach methods, we can get a better grip on that. Not only cell and tissue culture models, but also the in silico, the mathematical models, the artificial intelligence, it all helps. So we have been working 3 years on standardization. I worked a lot also in the field of neurotoxicity and development neurotoxicity. At OECD now, they're working very, very hard to continue the work we have been starting with many years ago to get, let's say, these methods into test lines, because the developmental neurotoxicity methods now are done with rodents. And as you can imagine, these in vivo methods, they might to a certain extent be predictive, but for these individual effects, it gets very difficult. So I think now Thomas has been speaking, so it's very, very important to develop microphysiological systems for use as regulatory tools, to look at a lot of these biomedical and life science challenges we have now with being exposed to many different types of external insults, be it pathogens or be it also chemicals and specific materials. So as you can see, stem cells, stem cell derived brain liver chips, looking at brain permeation, people, these are very recent publications. The field has been booming. And I think we are in a very good time now to really start to use these methods. And this is something that I've been working a lot on to try with many others to get these test methods in a toolbox, also at OECD and also in the companies. And these methods basically are really, in a way, you can simplify any of these methods, even the microphysiological systems. You have a test system, it can be different cells. You have endpoints or markers to measure, and you predict something on that. Important is that what you use there is reproducible. And secondly, it's relevant. It has a scientific basis. And this relevance also translates in predictive capacity. We talked about the liver, and the liver is very close to my heart because a liver already designed, we published all together in 2006. We need to have this biotransformation machinery as well as when you especially work in toxicology to understand when compounds entering the bodies, what are they metabolites? But also we need many other methods, what barriers they cross. So we need ADME methods for absorption, distribution, metabolism, and excretion. So we have been showing that these methods, metabolism methods, quite complex methods, which, for instance, have deep underlying molecular mechanism like induction, you can standardize them. You can standard human hepatocytes quite easily, cryopreserved. You can standardize the new generation of, cells that are available, like HRG cells. And so we have been investing for the last year a lot on analytical methods, toxicokinetics, and metabolism to get all these tools in the toolbox we need for standardization. So to come to this part of my talk, global harmonization is really important for these microphysiological systems to really have a better response and preparedness for health crisis that we're facing now. So what we have seen, and I think David will speak about this also, is that unfortunately, and this is not only with life science, but in general, science has an issue of reproducibility, reliability. And often this is due to better design of methods, better supervision, looking across labs that you can reproduce these methods. incentives for better practice, external lab validation, journals enforcing standards, I would say organizations enforcing standards. And this is something which is still work to be done, not finalized. We have been working with many to get this good in vitro method practice guidance at OECD. And today in this session, we will talk together on GCCPs, GCG giving, and GLPs. But in a way, putting all this together, we want to have these methods be used and increase the trust and credibility of mechanistic methods and the related data, because we believe that this is the only way forward to get really, for instance, methods for better understanding the complexity of human biology and the diverse reactions we have in terms of efficacy and safety. And so that's why we badly need these methods for regulatory purposes. So with many of us, we have been working to get, this information out in the public domain. And also with Thomas and others, we have been working on aspects like evidence-based toxicology, but the same thing you do with regard to evidence-based efficacy. So we started this journey in 2005 on good cell culture practices. David will speak a little bit more on that. We all still continue to work together, incredible but true. So, but it goes very slow. So one of the things that and others think, how we can amplify, how we go quicker. We have been trying to put our money on global harmonization, and it definitely helped a lot to get these methods better standardized. We are continuing to use our work to put it, for instance, in Europe, in let's say, systems like UCLIP, which is one of the systems that the European Chemicals Authority Agency is using, and also the Food Safety Agency when new methods have been introduced. So when you look at this document, it's about roles and responsibilities. We all want to do good science. It's about understanding and know what you're working with. It's about using standard operating procedures. It's about looking at the performance of these methods. It is about the nitty-gritty details of good scientific practices and good quality practices. So it's a bit like, historical people, like Leonardo da Vinci. Before you start to design something, you need to see if it works, in practice. So I think with all this complexity now of these methods, I strongly believe that any complex methods can be standardized, can be reproduced, but you need to know what you're working with. And this is the principles of this good cell culture practice. So it's not about, of course, when you have a simple 2D model and you have more complex methods like spheroids and 3D methods and microphysiological systems, it takes more time to standardize. But also technology and measurement methods have been boosting there. And if you look at the cryobiology, and you can do cryobiology under good manufacturing practices, you can have skin models under good manufacturing practices. You can do it also with microphysiological systems. What is the essence of all this? You need to know, and you need to know what you're working. And these are a few questions, and the slides are all available. If you work with cells and tissues, you need to first question why that particular test system, why it's relevant. Where do I get it? Is there a good supplier which works under quality control conditions? Do I need some extra characterization to know what I'm working with? Does this test system has metabolism component, because it seems to be very important? If you work with cells, do you know something how long they're good? Do I know something about the complete composition of media and reagents? Are there undefined media ingredients like fetal bovine serum or other ill-defined animal-derived things? What about my apparatus? Is this an apparatus that is, do I do the right checks for it? What about if I have a method, you need some kind of controls. If I have compounds, you know, the test items, do I know something about? Are they stable? Are they soluble? You know, do I have those range finding? When I get the data, how I collect the data, how I analyze them from very simple data to very complex data, you know? So,
Sandra, this
so
was a
whatever you use,
reminder.
5 minutes.
Yeah.
Okay, I'm 5 minutes, I'm over. Thanks to remind me. So stem cells. I mean, people have been working now a lot with stem cells. We have been also publishing on this already with Laura Suter, etc. So whatever test system you're using, stem cells, three-dimensional systems, spheroids, you can standardize these methods. I truly believe that. And so we have been, I'm near the end of my career. I started to wonder that publishing all these things, does that speed it up or not? Do we need to do something else to really get these methods used? And I think truly that something else happened. And by magic, they started to use these methods because it was the only resort to get some answers. And that was, I say to my kids and to my family, with every life challenge, with every health challenge we have in our family, there comes a gift and opportunity. And besides the great suffering that COVID-19 has been caused in the world, it also allowed to finally start using these methods for great health challenges. So together with people over the globe, so also with Surat, with people in South America, with people in Italy, medical doctors that were, Italy has been really had a lot of people in the beginning of the COVID-19 pandemic, lots of death cases because we didn't understand the disease, it was in the beginning. And so I worked also with medical doctors. We had the opportunity to have studies, also pathology studies, cadaver studies. So what we did, we put all our heads together. And we had a big project also at the GRC, it's called CHOW. Several are involved in that. And suddenly they started to use all these new approach methods, organ on a chips, spheroids, you name it, to better understand COVID-19. And here you see the list that we put in our publication. It's now a book. There are 2 volumes. This is the book we have been contributing with our chapter on knowledge for human relevant cell tissue and mathematics methods as key tools for understanding. But you see, they suddenly start to use all these methods, They start to use organ explants, dissociated cells, 2D, 3D, organ on a chip, whatever. So also at the European Commission, this is my colleague Ana Ruiz. We are leading a portfolio on better preparedness and response to health crisis. I think also there for One Health, for issues related to zoonoses, to issues related to chemical insults, I strongly believe that all these methods you're discussing and learning about by all these people that are sharing their knowledge are the methods to better understand the complexity of systems biology for a One Health approach. So these are some of the last works we are doing, mainly on COVID-19, but also on health technologies and innovation. This is one of the papers that we published, I think two weeks ago. It came into the public domain. This is using all these alternative methods, these new approach methods to understand, for instance, short-term anosmia in COVID-19 and using the adverse outcome pathway framework for doing that. We have other work ongoing also on long COVID, on neurological effects. Elena Hochberg, our paper came out in Cells. So I just hope that I inspired you with this talk of 35 minutes to show my personal story to say that we need to work together. But one of the things, I think it needs to go much faster than we have been doing the last 3 decennia to get really these methods used. And in a way, with although also lots of suffering, I can imagine in your families with COVID-19, like I say to my kids, if a great challenge happens, there is a gift and opportunity. So I think finally, new doors are opening to better use all these investments we have been doing with so many over the last years to use these no approach methods. I share with you this Twitter picture. I think it's so beautiful. These are spheroids, and since the chair is Armin, I thought Armin, I show this beautiful spheroid tweet that I saw on Twitter. So I think social media, I don't like this social media, but this is another avenue to speak about our work. So I recently started to use this. I think we can help there also to transdisciplinary, make people aware about the beauty and the need for these types of methods you're discussing all week. So I'm very grateful for the whole team that invited me. It's really a pity that I cannot be there with you 1 week, but I talked with many of you already over LinkedIn and Twitter, and I hope we can think together how we can better amplify the use of these methods now to stop using, needless animal methods that don't give us the answer for the really complex things we have to deal with. So thank you all so much for your attention, and thanks so much for the organizers for allowing this fantastic EMBO course to be face-to-face.
Thank you, Sandra. Thank you for sharing your private life with us and also your career steps you made. And I think you are a role model for an emancipated woman, which is a mother, but also a successful scientist. So that's I think that helps also our younger colleague to see how this is possible to combine it. So now I'm open for questions.
Hi, sorry. You got me in with one particular slide where you had mentioned people really struggle to reproduce the data of other scientists, and 70% of them indeed fail to do so. I was just thinking about the reasons for that, and one thing that came into my mind is whenever you read a manuscript or a paper, the most ignored or the part of that paper which is not very well written or is not very detailed is the methods and materials section. So do you think the publishers or as scientists we should do something about it to be more open about what practices do we use in our lab? Because what we do in our lab as cell culture might be completely or a bit different from what people do elsewhere. And these nuances perhaps might be the reason why we are not able to reproduce major or critical data. Do you think we as scientists have something to do about it, maybe publish only detailed methods papers, or I mean there are certain publishers which do that, but they are very confined to very specific methods, and as for microphysiological systems, the systems get so complex, you have multiple methods that one uses, and is there where we can all get together to do something about it?
I think this is really a fantastic question, because I totally agree. And my colleagues at the GRC and also others, and we will be doing something, actually a kind of theater play at the next ESTIF conference in Barcelona. So Sophia Leite and others, we had 2 weeks ago, we had, let's say, look into this area of academic data. And what are some of the issues and what paper she has done. Sophia Leite has done a great work also to look at what journals, you can publish much better on your methods. I think, there, like in everything, it should come from bottom up, top down. So the journals themselves need to have something quite easy in supplementary material, even if they don't want, I mean, you can say the space in let's say the cortex is shortened, but we are now publishing also on a framework. You know, you put all the stuff like the given criteria, you put them in the supplementary material and the materials and methods. But that means if you are looking and you want to use artificial intelligence approaches, you know, the supplementary material should be in the main information section when you want to automate. So there is a lot of work to do. And I think you as researchers definitely could try to find those papers, and I can send also the material of the hard work of my colleagues in the GRC and all these sources they have via Sophia Light and others to you as organizers to distribute to all those that were on the course to really publish in these papers where you can have more materials and methods, because the tragedy is vast. I work with EFSA, European Food Safety Agency, and I've been doing a risk assessment, a very strategic one. I've been involved to see how I could better embrace artificial intelligence to amplify what we're doing. Now, the tragedy is like that. With all the in vitro cell and tissue papers that came for that compound that I was analyzing in parallel with another assessor, almost 70% of the papers, even more, cell and tissue culture papers that were used to look at endocrine disrupting effects, because that's the area I work now most on the last year. So estrogen, androgen, thyroid, and steroidogenesis interference. And I've been working and a lot now on thyroid methods. Most of this public data, academic data published, we cannot use these methods. So we cannot do risk assessment because most you have no information. Most what is published has a high risk of bias. So it starts with the method information. And this, I'm so happy that this message came out of my presentation now, because this is the key message. But big journals, they try to do a lot, but not enough, because we still use old data. So then you have to go back to these developers of 20 years ago. But you know, you can better start late than never. So I think if you want to do something, it's like customers drive the market. If many of us don't want to publish in papers where you don't have methodological details, we can change a world. So I think your suggestion is really, really super important.
Thank you so much.
Hello.
Nice to see you, because
So, ma'am, a very basic question. A lab who is already into in vitro services and is also following good laboratory practices. So, how is this guidance document on good in vitro method practices is going to help them? Because being from GLP and following in vitro assays, so we need to take care of each and every components that we include for any in vitro experiments. So, how is this guidance document going to help them?
Yeah. So you're a CRO, probably, yeah? You're a contract research organization?
Yes, ma'am.
Okay, I think that's another. So, I mean, I talked about academia. I think industries, because I've been working all my life and still work with industries, because I think all stakeholders are critically important. One of the important things I started in industry is that basically, you need to work under GLP in some sectors, or you cannot get your new chemical entities on the market. So the quality part is already completely done. And I assume you follow also the OECD guidance advisory document 14 on GLP and in vitro, right. The one we have been issuing, exactly. Now, I showed one slide where you saw these three balls, GCCP, GIVIM, and GLP. They're not completely overlapping. So the thing is that you can do a perfect GLP study, compliant, but that the results have no relevance because the design of the study is not well. So often you use in vitro methods with OECD guidelines, that's guidelines on in vitro, and they're done well, yeah, because they're described. But many, I think we need much more methods. So the good in vitro method practice, and also some papers from Adler et al., which came out of the Orion cluster I think I put it in my list of publications. They look more at the relevance of the experimental design. So if you saw this slide on the GiveImp where I said all the things that you have, 66 criteria actually we have. And I can share that also with Surat. It's a template where you have to look into 66. We're putting that in the public domain now. It's already at the Euclid system as a help text. All you have to look at. And this means that only doing GLP is not enough. You need really to look at the science behind your methods, the science behind the test systems. Sometimes you see also in these papers where I did the analysis for EFSA that, you know, they use methods or test system totally irrelevant. And sometimes they use the good ones, but then maybe they don't do a stability test for their compound. And you know, this compound is not stable over time. So let's say the experimental design, the science is so important. And this is you as scientists who know so much more. You need to really, Thomas Harting always said, one of the first things we said in the GCP and David will be speaking about, know what you're working with. And the only ones that can know what you're working with are you as scientists, but also in your companies, you know. I think everybody's a scientist because what does a scientist? You explore, you innovate, you navigate, and you do then laser-focused action. You know, that is our praxis. So we need to be sure what we're using we can use, because sometimes we use things that you just cannot control, that are so wiggly, that are drifting, genetic drift and things like that. Therefore, good manufacturing practices for cell systems. That's why these tissue systems, they were so successful because they were produced by companies that worked, a lot of them under GMP. So all these elements from a test method, you should standardize. Okay. So besides the GLPs, you need the GiveImps and the GCCPs. I mean, the GCCPs are part of the GiveImps. Yeah, David will show that also.
Is it okay? Have you got your answer?
Yes, yes.
Yes, we have time for one question.
Yeah.
Then
Thank you,
we have
ma'am.
to go into the break.
Hi, Dr. Sandra. It's a very great and encouraging talk. Thank you so much. I have a just quick question because we know human brain is not like a mouse brain. As we have discussed 4 days, human is not 70 kg mouse. And how do you convert the neurotoxicity assay? Because so far in our knowledge, there is no OECD guideline which advises us to do human neurotoxicity. What's your suggestion? And is there any OECD guidelines coming up? It's in the preparation of pipeline?
Thank you for a very important question again. I mean, they are now doing a lot. Anna Price, my colleague, Lena Hochberg, Magda and so on from OECD. There is a big effort now at OECD on working on developmental neurotoxicity. The problem is, when you look at the toolbox we need, it are many, many methods. And there is definitely not a huge set of methods. With the paper that I've been showing, like on anosmia and the combination with the adverse outcome pathway, you have more methods. We discussed also about neurological effects. You know, I'm in a group at OECD. We had meeting this week on, let's say, thyroid disruption, you know, endocrine disruptor. Now, obviously, if you look at the literature there, the levels of T3 and T4, I mean, they are linked to neurodevelopmental effects and so on. So the complexity is so big that you need integrated approaches. So I think they are trying to do a lot to bring as many of these methods into the OECD toolbox, but it will take years because the speed before you get a test guideline, and we have been as EGVAM working on many of these methods, it takes a long time. So in the meantime, we need to start using the GiveImps, yeah, to get your methods as standardized as possible. And so I strongly believe that the area of neurotoxicity or neurodevelopment, neurobiology is important. But already, if you look at our paper on anosmia, For instance, to deal with anosmia, we don't have in vitro models. You know, if you think about olfactory bulb, where you have the sustentacular cells and you have a direct connection with the brain, and many of the effects, you know, of SARS-CoV-2 are linked. It's very important. I like a lot this concept, combining the clinicians' great knowledge with the adverse outcome pathway understanding of what happens. So I think if you look to this anosmia paper, we need many methods, not only one method. So we need a toolbox. And I think researchers embracing this kind of mechanistic understanding and mechanistic systems biology, and that they design under GIVIMP the good methods they need, there will be a way to use these methods. Because, for instance, EFSA is using already the methods. They're not yet OECD test guidelines, but when I'm doing the analysis, you know, if these methods have very few bias and are compliant with GIVIM, for instance, we use them even in tier 2 risk assessment. Of course, now we only use the GLP studies because these are the only ones that go into tier 2. All the rest that I saw in the public domain went into tier 3 and tier 4, high risk of bias. So I think by working according to high standards, although that there are no test guidelines, we can move a world. I mean, that's really what I'm.
Yeah. Thank
Sandra,
you.
last,
Thank you.
For action.
Thank you.
last word, yeah? That's it? You got your answer? Yes. Okay, great. Thank you, Sandra. We are ready to make a break, so thank you very much for this beautiful presentation. So, and now we have a short break this time. So thank you again. Applause for Sandra.
Applause for all of you and for the work you're doing all together. Really, I thank all those that already reached out to me via social, and I hope to be in contact with many of you. We are stronger together.
Thank you very much.
Thank you so much. So I leave now the meeting, or how does it work? Or yes, probably yes, eh? Arin? I cannot follow the rest, right?
I can't hear you acoustically. Yes,
Can
please.
I follow?
Thank you. Now it's a break. Thank you. Thank you again. We stop here. Yeah. Thanks again.
Thank you, Dr. Wolf, for chairing the session. Thank you, everyone.